Okay, so in today's lesson, we're going to actually keep talking about making proteins, but specifically we're going to talk about how our bodies actually regulate genes, so stretches of DNA that make proteins, so that we can actually turn genes on and off. So technically, we're controlling the manufacture of protein, okay? Gene regulation is what the general term that living things do this naturally. Us humans, we're able to actually do this nowadays through what we call gene therapy, which is pretty cool. Have a go at the three quick questions. Pause the video here and we're going to go through them together right now. Alright, so hopefully you worked out that this structure here that starts off as a double helix but then gets unzipped is the DNA. More specifically, it's the DNA template because it's getting copied by the mRNA, well, by RNA polymerase into mRNA, which is this structure here. This is the mRNA or the messenger RNA leaving the nucleus, so most likely it's a mature, well not most likely, certainly it's the mature messenger RNA. All right. Number 3 here, we've got this whole structure here, and this whole structure is the anticodon here, anticodon. Right, and the anticodon is attached to the transfer RNA, the tRNA, right? And lastly, we have an amino acid stuck to the tRNA, right? And then we've got down number 6, we've got on the mRNA, we've got the codon, which is complementary to that anticodon, okay? Number 7, we've got these strands of amino acids connected together by this bond called a peptide bond. So this must be a polypeptide chain. Okay? All right. And then lastly here we've got this big awkward looking structure which is just simply a ribosome. Ribosome made up of ribosomal RNA, right? Okay, I'm not going to go through the role of each of those. All right, you should know the role of each of these. If you don't, have a look at Google and your textbook or Biozone, and they should go through a few of those roles. If you have any questions, let me know later on. Okay. All right. So here we have a variety of proteins can be manufactured from a single DNA sequence. Describes 2 ways which this is possible. All right. So the 2 ways are, so if this is transcription. Right, we can have post-transcriptional modifications. So that's modifications to this molecule, the mRNA, in the process of going from pre-mRNA to mature mRNA. All right, it can go through the splicing process. Right, so for example, let's say there was here when we're actually transcribing, there's introns, so sections that are going to stay in the nucleus, introns there and introns there, right? So the cell can decide, okay, so I actually want to keep those introns in. So we're going to transcribe and translate this whole section, right? Or the cell can decide, oh, I'm going to pull out this part, right? So I'm only going to transcribe, so only the exons are those sections there. Or the cell can pull out this part, or it can pull out both, right? So from that same sequence of DNA, we can get different messages. Okay, similar messages, but different ones. But similar messages are going to end up having completely different proteins regardless. Okay, the second way that we can actually make changes is through the second process, which is translation. All right, after translation, once this polypeptide chain has left the ribosome or left the endoplasmic reticulum, we can go some post-translational modifications. So that can be things like the building of disulfide bridges, or there can be things like methylations. Okay, don't have to know any of those specific ones, you just have to know they're called post-translational modifications. Okay, all right, so today we're going to talk about gene structure and regulation still, but firstly, we're going to make the distinction between what we call structural and regulatory genes, and then we're going to use a really specific example of the lac operon, and you have to know this one specifically, which is a simple prokaryotic model that illustrates what we call the switching off and on of genes by proteins. Okay, and these things are called transcriptional factors, and these are expressed by these regulatory genes that we're going to talk about up here. All right, so how do cells talk about turning genes on and off? So cells actually have two different kinds of genes inside of our DNA, and the first kind is called these regulator or regulatory genes, all right? And what these genes are is they're genes that transcribe and form RNA and then later on proteins that are called regulatory proteins, and they have the ability to actually alter the expression of other genes. Whenever you see this term expression of other genes, what we're talking about is the reading of genes to make some proteins, okay? So these regulatory genes make proteins that control the making of other proteins, essentially, okay? Structural genes, on the other hand, like this little blue one over here, right? They're any gene that don't actually do this. Okay, they are simply genes that code for normal proteins. They can be enzymes, they can be hemoglobin, immune antibodies. Gosh, really struggling here. But lots and lots of different ones, but they just don't control the expression of other genes. Okay, another important thing to point out is this operator. Okay, I want you to have a look at this because it's going to be important a little bit later on. Okay, so let's talk about a short story of a very important bacteria, and that's this guy here, this guy called Escherichia coli. Okay, so as mammals, we're not actually able to break down the sugar component of milk called lactose, and so we're actually helped out by this bacteria called E. coli, and they live inside our small intestine. E. coli actually produces proteins, more specifically an enzymes that can break down lactose into simpler sugars for us. Okay, so an important thing here is that if E. coli actually constantly produce these proteins, even if I haven't consumed milk, it would quickly run out of energy. Making proteins takes a lot of energy. So E. coli actually needs a way to regulate the expression of the genes which code for these specific proteins for the purpose of saving and conserving energy. So that's where this thing called the lac operon comes in, and the lac operon is inside the DNA of this E. coli bacteria. Okay, when the lac operon is turned off, a specific regulatory gene called the LAC-I, okay, it produces mRNA, and this mRNA is translated to produce a protein, all right, which is called a repressor protein. And this repressor protein, it has a very interesting shape because the repressor protein in this form, in the active form, fits perfectly into this operator we were talking about. All right, and this operator is right in front of the structural genes. All right, and there's those structural genes. So that means that when RNA polymerase goes to read this strand of DNA, it's going to read that regulatory gene, make the protein, but then it's going to keep going along, and as soon as it gets to the operator, it's going to be physically stopped. So it's not going to be able to go and transcribe those structural genes, okay? So this happens when there is no lactose present. So if lactose is absent, the lac operon is turned off. Now, imagine you have a huge big drink of milk, okay? 90 seconds later, all that lactose, delicious lactose, reaches your small intestine and swarms of these E. coli bacteria. And so, now that lactose is present, the lac operon is going to turn on, okay? So what do we mean by this? So RNA polymerase still reads the regulatory gene, LAC-I, okay, producing that repressor protein. Okay, this time though, lactose, which is represented here in green, okay, that's lactose, goes into that protein, okay, because it fits snugly into this protein, and by fitting into this protein, that protein undergoes what we call a conformational change, which means just that its shape has changed. Why is that important? Because remember that this side over here of the repressor protein, it fit really nice into the operator. Now look, it won't fit anymore. Now that it doesn't fit, RNA polymerase can go and run down the DNA, doing transcription, that transcribed bit of mRNA can go and be translated and form these 3 different enzymes, which you don't have to know the name of. But the important thing about these 3 different enzymes is simply that they're the 3 proteins that work to break down the lactose, okay, in our bodies, okay? So as this lactose is actually broken down, the concentration of the lactose is going to go down, okay? Less lactose is going to be present now to bind to these repressor proteins, okay? So the lactose is going to actually pop out of these repressor proteins, and once again, the repressor proteins are going to fit into that operator, all right? And so this process that we just talked about here, where we're getting rid of all the lactose because we're producing these enzymes which are able to get rid of the lactose, it's reversible, okay? We're going to regain some repressors, and they're all going to fit nicely into here, which means that now we once again have the lacqueron turned off. Okay, so we can regulate now the production of these 3 proteins, okay?